Hello everyone, I'm Junya Hayashida from Kyushu University. Today, I'm going to be talking about simple but powerful cell tracking method. This is a joint work with Kazuya Nishimura and Ryoma Bise. First, I'd like to explain the difference between ours and the previous approach. State-of-the-art methods provide tracking by independently estimating cell position and motion. In this approach, there may be incoherence between detection and association. It may affect the tracking performance. In contrast, we perform tracking by jointly estimating the cell position and motion. Joint estimation allows us to take advantage of the spatial temporal context and guarantee coherence between detection and association. We propose a representation that jointly represents both cell motion and position, named Motion and Position Map, MPM. In order to explain MPM, I'd like to first explain how to represent each cell motion. As shown in the figure, when the image sequence is considered as 2D plus T data, the position of the same cell is given as 2 points as an annotation. This annotation contains both cell detection and association information, and can be represented by a starting point and vector connecting 2 points. Therefore, as in this arrow, the motion is represented by a 3D vector from frame T to T-1 and embedded on the pixel of frame T. And this is also embedded around the cell position at frame T. The motion vector field thus obtained is a representation of each cell motion. Next, I'd like to explain the representation of the cell position. The cell position is represented by such a likelihood map. As shown in the figure, the likelihood map is a Gaussian distribution, with the cell position annotation being the peak. Here is an overview of MPM. The motion vector field and the likelihood map are combined to the motion and position map, MPM. In MPM, the direction of each 3D vector is the cell motion, and the magnitude distribution is the likelihood map. That is, by exploiting the direction and magnitude of 3D vectors, MPM jointly represents the features related to detection and association. Next, I'd like to explain the outline of cell tracking using MPM. This method is very simple. First, the MPM is estimated by CNN from 2 successive frames of input. Then, from the likelihood map, which is the magnitude of the MPM, the cell positions can be localized by detecting the peaks. Then, the cell motions can be captured from the vectors stored in the detected positions. Moreover, during mitosis, each of these 2 vectors face the same direction, so mitosis detection is also possible. In this way, cell tracking can be easily performed with MPM estimation. In the experiment, we use an open-ended set of phase contrast microscopy. This dataset consists of hundreds of mouse myoblasts cells, each with a different appearance in 4 conditions. Here is an example of the estimated MPM. Like this, MPMs are estimated for each frame pair. Here are the tracking results under 4 conditions. It was confirmed that our method works well in each conditions. This table compares the association accuracy. As a result of quantitative evaluation, our method achieves state-of-the-art under various conditions. In summary, we propose motion and position map, MPM, that jointly represents both detection and association. And in the experiments, our method outperformed the state-of-the-art method under various conditions. Thank you for listening.